Hi everyone, welcome to this episode of Keipert Labs. In today's video we're going to be discussing the concept of DNA replication. Okay, so we've been thinking in recently about deoxyribonucleic acid, also known as DNA. Okay, so DNA. Okay, and we're seeing that it's a molecule that's got this ladder-like structure. Okay, that the backbone or the sides are made up of alternating units of sugar called deoxyribose and phosphates connecting them together. And then the rungs are pairs of these chemicals called bases. These bases, so pairs of bases, so kind of working in this sort of paired sort of arrangement. So we have four bases, A, T, C and G, which are adenine, thymine, cytosine, and guanine. But we just give them, we stick with those letters for their code, for short. Okay? And then what we see is that we always get particular pairings, A and T and C and G together. But now one of the ideas that we encountered a little bit earlier on was that the fact that your cells in mitosis in particular, but also in meiosis, we need to make copies of the chromosomes that exist inside that cell to make new versions. So today we're going to be doing a little exercise to illustrate what that looks like. And so I'm going to make use of some colored little buttons to help you picture how this works. Okay, so what I've got here is I'm using these magnetic buttons to represent the base pairs of a DNA strand. I've established a bit of a code here. Okay, so if and these are the sides of the ladder here. Remembering that A and T and C and G always pair together. Okay, so if I'll go through just quickly and write out what the code would be, just to think about these letters together. Okay, so we've got C and G, A, T, G, C, T, like this. So that if we were to write it out as letters, that's what I would be writing. But for now, we're just going to stick with colors so that you can kind of see how this process works. Okay, so what we need to be able to do is we need to end up with another strand that's exactly like that. Okay, and inside your cells, they've got a really particular way that this happens called replication. Okay, so replication is making exact copies, is copying, making replicates or kind of exact copies. Okay, that's where we get the word replica from. Okay, so what happens? The first step is that the strand unzips. Okay, so in here, it's actually like I've got them all pressed together, like they're kind of locked in, but actually it's a little bit more it's a little bit of a gentler kind of locking in that happens between here. Chemically speaking, it's not as strong as the rest of the connections in this molecule. So what it does is that then there is a particular enzyme, a particular chemical here called DNA polymerase, that is responsible for traveling along this strand and unzipping or separating the strands from each other, pair by pair, traveling along like a zipper, kind of travels down where the metal teeth kind of connect together and then pulls them apart. So what we end up with is we end up with these two separate half strands. Okay, and then what we do is that then these get built from, so that we end up with exact copies based on building off these as a template, because we know that A and T and C and G always pair together, and I'll even write that just over here to remind you, so that we can know that if I have an A, then there must be a T. If I have a G, then there must be a C. So what happens in the next stage is that these little kind of base kind of sets, which we call nucleotides, then kind of start to build up the rest of the strand. So knowing that this one has to pair with green, has to pair with a blue, then that one can add on, and then this one adds on, this one adds on, this one, and then this one. And then I get exactly the same thing. Well, my board's just shifted. But building the complementary kind of strand. So we get different bases that now come along and connect up to the gaps over here. But the effect is exactly the same. So we've added in the complementary nucleotides. Okay, so they're the ones that that fit together. So not the ones are the same. We didn't find greens to connect with greens and blues to connect with blues, but we found their partner. Okay, and now what we have, if I shift this down just a notch, is that I now have two identical copies. Okay, so I've taken one strand, unzipping through the middle, using with this special chemical which does the unzipping, and then adding on the nucleotides to fill in the spaces, to pair up with the right things, and then we end up with two exact copies of the strand, or the DNA kind of string molecule. And so now this one can go in, like if we're thinking about mitosis, then this one can go one way into one cell, this one can go the other way into a different cell. And then from there. And so then in the next cell, if that cell needs to divide, we go through the same process of unzipping and then building up the complementary nucleotides, or kind of adding on those bases to fill in the spaces. Nice rhyme there, Mr. Keipert. To then end up with more copies, more and more copies. Every time, every strand, we end up with two lots. Okay, so the more, so every time it doubles. Okay, thanks very much for watching. Bye for now.